He comes for all the talk, soon he can okay. Huh? I think so.
Okay, welcome everybody. Let's get started. It's a great pleasure to have Professor P.B. Sunil Kumar here to deliver this colloquium on chromatin folding and condensation. Sunil has a very long association with RRI, so I'm not going to give a long introduction. He did his PhD here on liquid crystals, and after that he's moved to membrane biology and other stuff, working with Madan and other people. And he is before going to, while he was at IIT Madras, he also took a stint at IIT Palakkad, setting it up as its first director. Hugely responsible task of building the whole place up. And now he is back at IIT Madras, and he is doing a sabbatical here for 1 year. So Sunil is going to be around, so anybody who wants to catch him, especially students, to discuss various aspects of soft matter physics, please take advantage of his presence here. And with that short introduction, I invite Sunil to give the colloquium.
Thank you, Pramod, for the introduction, and thank you for asking me to give this talk. So I'm going to talk about some recent work which we did on chromatin folding, modeling that. Since this area, I don't know how many of you are familiar with, so it will be a slightly long introduction, so in the spirit of a colloquium. And so this work is done with three people here. What I'm going to present today. Saudamani Sahu was a postdoc with us, and she's now in NIT Rourkela as a faculty. And Ranjit in IIT Bombay, and his student Sangram Kadam, who is right now in New York University. So as a postdoc. So this is the collaborators, and the work was funded by NSM DST. Okay, so let's get started, and feel free to stop me if you have something to ask. Okay. So you all know what DNA is, right? Okay, so and it's a long string. Okay, so the first problem so I wanted to introduce what are the big problems and which one is trying to address, and then go into smaller aspects which we look at. So the first problem which people talk about is a packaging problem, right? That it is a very long DNA packaged in a tiny cell nucleus. Okay, so we are talking about a 10 micron size nucleus and about 2 meter long DNA, and it is packed into that. So there's a 1,000-fold or 10,000-fold packaging issue. Okay, so this is one problem. So how is this packaged, right? So that is one question which people try to answer. And other issue is an information management problem. So that is that there are different cell types, okay, but we have the same DNA for all the cells, right? And different cells do different things. So obviously, even though the DNA is coded, okay, different cells read off different things, right? So how is that done? Okay. So for example, if you could have the cells in the skin and the cells in our eye, they would look very similar. Okay, and as far as the DNA is concerned, they are symbolically showing that the DNA is the same in both of them. Okay, but they are doing different things, we know, right? So, so that is one. So how do they manage? How do they show this behavior? Is one of the information management. Okay, so there is information both of them, but some different information is read in different cells. Okay, okay. So that is what we call the epigenetics, right? So that is one issue. Okay? So the answer to that is that cell DNA in the cell is not bare. Okay, it is covered by a large number of proteins. Okay, and we call them as chromatin. Okay, so DNA covered with protein, we call it as chromatin. Okay, and that is the basic answer to this question. Okay, so this protein coverage and the packaging could be quite different in different cells. So basically saying that DNA is assembled differently in different cell types as chromatin. Okay. And so even though the DNA is the same, they're kind of packaged differently, okay, in the different cells, so they behave differently. So that is one issue, okay. So so that's the bottom line. So there is basically chromatin organization is, DNA self-assembly is different in different cells, cell types. Okay, now the question there is then how is the chromatin actually organized? We know that, okay, they are organized differently, we say, okay, and that is the reason. Okay, now the question is how they are organized in 4 dimensions, right, like in time and space. Okay. Because we know also that it is dynamic, the cells behave differently in different time, it has to respond to signals, right, and cells in the eye has to produce a tear when it signal. Okay, so it can't be always doing the same thing, so there is also time dependent. Okay, so there is also a response to signal. Actually, Madan has a recent paper on that signaling issue. Okay, and so that's one issue. Another thing is when the cells divide, okay, we know that DNA is copied. Okay, but the question is how is this kind of epigenetic information, that is the one which is covered with protein, and something is suppressed, something is active in different cells, and how is that being inherited, this information. Okay. So these are big questions which is to be answered. Okay. And so that's a bigger motivation for doing a lot of this work. Okay. So let's see how this DNA is actually packaged, okay, for those of you who are not familiar, okay, and those of you who know, maybe, yeah.
Just to clarify, the epigenetics is the environmental part at some level. So that's why you're asking the question, how can that be inherited?
Yes, so genetic.
Does the DNA, you inherit just the DNA without this chromatin
Correct.
structure, is that what?
Yeah, so without the protein, if you only have the DNA, it's the same for all the cells, right? So DNA is copied, we
But
know.
also for inheritance,
Yes.
only that inheritance when you do meiosis or whatever,
Yeah.
then again the cell division that happens,
Yes.
mitosis, meiosis also, right?
Mitosis. Yeah.
Mitosis is for, whichever one that you do,
Mitosis
for
is
sexual division,
Correct.
right? So, there what gets inherited, we always assume is the DNA. But are you now suggesting that more than just the
More
DNA
than
gets
exactly.
inherited, including chromatin, is
Correct.
it? Okay.
Yeah. In fact, including the
That's-
structure in which it is.
I see, okay.
Yeah. Right. Okay. So that's again, I'm reiterating that here. So this is the kind of where it is packaged. So let's start with the nucleosome-level. Okay. So here is the DNA, double helix, right? And now this is then wrapped around a set of proteins called histones. Okay. And these histones are shown here schematically. And this about 150 base pairs with what it is wound around. Okay. And this histone, the string of these histones, okay, and then is then folded into another structure, a chromatin loop, is known as topologically associated domain. Okay, this is kind of about, it could be about 1 MB long, 1 mega base pair long. This could be, okay, and this typically, okay, 800 kb to about 1 MB. That's the size of these kind of domains. Now these domains are then further, okay, and packaged into further bigger domains, and basically what they call A and B compartments. Okay, the B compartments are the one which is not expressed. Okay, whatever is in the B compartment is not not expressed in the cell. Okay, and what is in the A compartment is more strongly expressed. Okay, so these are this compartmentalization, and we are talking about this compartmentalization of the size of a 3 mega base pairs. Okay. And then of course these compartments already assemble into a different higher level thing. It's called chromatin territories. Now these are different chromosomes. Okay, so during cell division, these chromosomes, in the nucleus, these chromosomes are kept in different territories. Okay, and there is small amount of intermixing, but there is not very heavy intermixing. And when the cells divide, even though the chromosomes condense, the chromatin condenses, and then they get copied, they get condensed, and then they get divided, okay, into 2 cells. But when the cells form, these territories are again restored. Okay, right? So that is a question of the whole. That is what the whole epigenetics is about.
This one part is expressed, right?
Yeah,
So
typically most expression happens in the A compartments. Yeah.
And so the part, so
part where it's expressed, basically this also there, I mean distance-wise they are closer, is it, in the whole DNA?
No. So what I meant by I think is that some part of the DNA is read off.
Yeah. So
right.
it's not like some part from this side and some part from that side is expressed in the same cell.
Yes, it could
So
be.
and then both part has to be there in A, or
Yes, so there are A compartments.
Okay.
Okay, A compartments, and A compartments are also distributed. Okay,
Yes.
it's not only in one place.
Ah,
Okay?
so it's not one A compartment where,
it's
okay, so there will be a lot of
distributed,
A compartments and
Right.
lot of B compartments. Okay.
So B compartments are typically towards the nuclear periphery. Okay, A compartments are typically more towards the center of the nucleus. Okay, so that is one thing, and also that this is more condensed. Okay, relatively. Okay, more condensed. So even though it's not, the densities are not very high, are very different. Okay, this is a more condensed and more tightly packed, and while this is a little bit more loosely packed. Okay, so.
In any one of these regions, once it comes in, does it go back in and out, or is it you sort of wrap it up and then you leave the region completely and then you wrap it
No,
up?
this is dynamic.
And also it leaves and comes back. So
Between
once-
A and B,
No,
that I don't
I mean, so it's a single strand, right?
Yeah.
That is now getting wrapped up in different.
yes.
And so if in some B region, you define the B region as something that it comes in once and leaves once, or is it that it can come in
No, it can be
once,
multiple.
leave,
It
and
can
then
be
come back into the same B
Remember,
region?
Yeah, right.
So very distant parts of the DNA can still be
In condensed
in
into
one region.
I will show
like
you
this.
that. I will show you that thing also. Yeah. Right.
So, regarding this packaging of this complex thing, so I was wondering whether these knots and all are also possible in this, or
Yeah. So there are knots are not very important issue here because there are this enzymes called topoisomerase which can actually cut and rejoin them.
Okay.
Okay. So yeah, that is not a implication.
To explain these arrows and what do you mean by topologically associating?
I'll come to that.
Yeah.
Yeah. Right. I don't know why it is called topologically associating, but I can tell you that it's a domain. Okay. Right. Yeah. So it is. We will see that in a minute. Yeah. Okay. So this is what it is. Okay. This package.
Same connector.
Right. Okay. Yeah. 1D connection. Right. Yeah. 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 Right. Because but that is also okay. Anyway. Yeah. So we'll come to that in a minute. Yeah. So it is. So this is a different level. This slide I wanted to show you is that there are this different level of packaging, and this packaging is what people are talking about as epigenetics. Okay. And because this is coming because of protein interactions and things like that. Okay. And these are what I mean by inherited is that when the cells divide, this gets transferred to the next one. Okay. So now how this is transferred is really not known, but but that is what people know that happens. Okay. So now let's go to the next thing. So the question is, before I go into, I think I just wanted to spend a few minutes, a couple of minutes on how does one experimentally measure this 3D organization, okay, because it's measurable, okay, and 3D organization in chromatin. Okay, so that is, there are many challenges, okay, in actually looking at the 3D organization of chromatin. And the main reason is that chromatin is not crystal-like, okay, so X-ray diffraction won't work, okay, and it's not very effective. Okay, and cryo-EM and staining methods have been used, but DNA is actually nucleus is quite crowded, okay, and contrast issues are there. But there have been works, okay, partial successes in actually looking at this thing using cryo-EM techniques, okay, and then there is most popular is what the people call as chromatin conformation and capture, or 3C, okay, and there are different variations of that at different resolutions called high C, micro C, etc. But basically it's called chromatin conformation capture, okay, and this been a technique of this century, and a lot of work which is done recently on that. Basically, I'll just tell you what this actually calculate. Okay, so then there is microscopy techniques. These are even more recent, where they use what they call a DNA paint technique. Okay. So now these techniques can do single cell, while here we are doing multi-cell average. Okay, these are getting really multi-cell average. Okay, but this is doing single cell, so it can also do dynamics. Okay. Anyway, some of these results I will show in the talk. Okay, So what does 3C or micro C measure? They measure basically the probability of contact between two segments X and Y. Okay, that's what it is. You have two segments. It could be one part of one string. Okay, as Madan said now, like it's like part of one string, but there are two segments of that string, and that contact between two segments. So for example, this is my DNA, long DNA. Okay, segment X, segment Y, and what is the probability of contact between these two segments? Different resolutions, typically about 30, 50 MB. KB. The resolution is what people look at these segments. Okay? Now this is what it measures. Okay, and then you can plot the probability of contact between two segments versus the genomic distance, or in polymer language we would call that as the contour length, right? The contour separation, right? The contour separation. Okay, and that you can measure. Okay, and then you can get a curve like that. And then you can look at different cell types and different variations of the cell, different stages of the cell, etc., and see how these contacts vary. Remember, these are many cell averages. Okay, so we are looking at the probability of contact between two segments. Okay. Well, because of this, I think there's an interest in polymer physics, because it's a polymer physics problem of asking of a string, and then you're asking the question that when is a contact between two regions separated by thing, right? So similar to a first passage kind of thing. Okay. And when it comes back first, and then like how long they'll stay in contact, various issues. When you're looking at really biological issues, if you want to answer, you should also answer the question that how long they stay and when contacts once, etc. But anyway, this is what we can measure in this experiment, and you please notice that these numbers are very small. Yeah. Okay. And that's the reason why you need really big averages to get this thing curves. Okay, and very many cells. Okay, and these numbers are very small, and hence this contact is extremely dynamic. Yeah, so it's very small and dynamic. Okay, so this is the probability of contact I measured in numbers. You can also plot it as a matrix, right? And now here we're showing this or the genomic distance. Okay, that is what it is. Different. And this axis also genomic distance, and you're looking at the contact between these two. So obviously it is symmetric. Yeah, it is symmetric axis. And now you see here, this red marks the larger the stronger the red color here. Okay, it is higher the contact. Okay, it is kind of normalized. Okay, and this plot, for example, is made with a 30 kb window. Each segment is about 30 kb. So now you can see these boxes here, right? Okay, these boxes you see, which means that these regions, okay, this segment here and this segment here has a very high contact, right, compared to other segments, and that is called the topologically associated domain, and that is a primary loop which is forming. Okay, so that is okay, and then you can see that this topologically associated domain, okay, has a weaker contact with this topologically associated domain, okay, and that is what is shown here, right? So the contacts decay as it goes. Right? So that's what you can see, and these are actually the different domains marked here. A, and then there is a smaller domain here, and then the C, etc. So that kind of makes it clear.
So experimentally, how do you count these contacts?
We can discuss that later.
Okay.
It's a long process, right? So yeah. So this is you do hybridization, and you have to cut it, and then you have to again form it, and you sequence the DNA, all of that stuff. We'll come to that later. Okay. So. Okay, so the typical resolution of this is, as I said, it could be 5 kb to 100 kb, okay, the resolution, depending upon what method you use, okay, and you can get this, basically this length of this things you're talking about, okay, average of that. Okay, so important points to remember about this measurement is that these experiments are population averaged, okay, averaged over many cells, okay. It cannot tell how a single cell chromatin looks like, okay. It cannot tell what the dynamic picture is, okay, but since the probability of contact is very small, the chromatin is highly dynamic. This is what we have to remember. Okay, in this case. Okay. But as I said, the DNA paint experiments that can actually look at single cell and then actually track this one also. Okay. And there are a few other things which you can look at the dynamics. Okay, so now the question is, how do chromatin domains form is a big question, right? So how do these domains form, first of all? Okay, that's one of the biggest questions you have. And there have been two popular hypotheses. Okay, and one is a loop extrusion model, okay, and in which you have a protein called cohesin, okay, it's an enzyme, okay, and this cohesin, okay, you can say, think about it as a motor protein, okay, and it can bind onto the chromatin, okay, and then it can extrude a loop, okay, and you have multiple cohesin binding and extruding these loops, okay, and then there are these other protein complexes called CTCF, okay, and this cohesin can go and it can, and this CTCF, and it can stop at the CTCF. Okay. There is also the thing that CTCF is not a hard stop; it can jump, okay, but more or less, because it's a complex of many proteins, okay, it kind of stops there, yeah. And now you can have many such things joining, and you can have loop on loop and things like that. Okay, you can have also my condensed thing, and that is one of the things I wanted to talk about here. Okay. And so that is one thing. Okay, another thing is called with phase separation things. Okay. And so I think Madan would be a better person to talk about it, because they had some experiments also recently, and in which these things happening, the phase separation here, saying that other proteins come and then bind onto the nucleosomes, okay, and those proteins induce interaction, and then they can form these domains, okay, and these domains, typically, if you want to say how these domains kind of come and then express and respond to signaling, etc. This is one of the models which people use. Okay? And there have been a lot of work on this method.
I have a kind of a nice question. So these proteins all don't have to be identical, right? The proteins,
yeah.
the histones or what?
Histones are, okay,
These are?
histone is a complex of 8.
The DNA and the protein.
No, histone is a protein.
Histone is a protein.
Complex, okay.
These are not histones.
No, there are 4 types of histones.
Okay.
There, okay, and it's histone octamer they form. Okay.
Okay.
And these octamer is form a complex, and the DNA winds around that.
Okay.
And now there are other DNA associated or
Okay. These are different
chromatin associated proteins. Okay, what we're calling here, what I'm showing here is HP1, for example. Okay, there are many such proteins. Okay, which even some of these things respond to signals also. Okay, and they can also come and bind. Okay, and there are also other enzymes which the histone I showed you in the earlier that it has a tail. Okay, and there are proteins which actually modifies the chemical state of histone. Okay, I will come to that if I get time later. Okay, and that can also change the interaction between these histones. Okay, this is nucleosomes. Okay, so nucleosome is histone plus DNA. Okay, and a nucleosome interaction can be changed by other proteins. Okay, which binds on them or modifies them. Okay, for example, you can acetylate or methylate and things like that, and then change the interaction and the structure of that. Okay, so that is all.
Do the probability of contact plot vary very much if the domain size changes, let's say from 5 kilobase to 500 and so on?
It's not really domain size dependent, but it also depends on the modification which it happens.
Okay.
Right. Yeah. Okay. So let me go ahead with this. So this is the 2 things. Okay. What I'm going to talk mostly about this. Okay. I would like to touch upon a little bit on this, but I may if I have the time. Okay. Otherwise, I'll be mostly talking about loop extrusion dynamics today. Yeah.
So when you write chromatin, you mean chromatin along with the DNA inside?
Yeah. So chromatin is DNA plus the
Plus
protein complex. Yeah. Okay.
So
Right.
things that the DNA has bound.
Correct. Yeah. So that's what chromatin. Yes. So what I'm showing here as a string is actually chromatin. Okay. It has everything in it. Yeah. Okay. And so okay. So let me go ahead. Okay. So we looked at 2 specific aspects. Okay. One is the effect of chemical change. Okay. Or change in the chemical state, as I just now mentioned. Okay. Induced by chromosome remodeling proteins. Okay. On the interaction between chromatin elements and the resulting organization. What can happen to that? Okay. And then the other one is the competition between time scales related to active loop extrusion and thermal relaxation of chromatin polymer. So basically, as a polymer thing. Okay, and that is what that appeared in this paper, the journal here. I'm mostly going to talk about this today. Here. Okay, so let me go into a little bit more detail on the model. Okay, so this is what we want to look at: the interplay between the loop extrusion mechanism and intrachromatin interactions and relaxation process which are associated with that. Okay, as a typical polymer thing. So I'm showing here the chromatin beads. Okay, here. Okay, so each of this bead actually represents 2 kb. So it's not one nucleosome. Okay, I'm showing now. Think about whole chromatin as just as a string. Okay, and I'm representing each 2 kb segment as a bead here in the model. Okay. So, and because of that, I will also talk about this polymer as a flexible polymer, because typically the chromatin persistence length is of the order of 200 base pairs, okay, and so 200, 250 base pairs, and we are talking here about 2 kb each bead is, so it's kind of flexible in that scale. Okay, so it's a flexible polymer. And then you have this cohesin. As I said, this cohesin is a complex which can bind onto the chromatin filament, okay, and then extrude a loop, right? And now, how we model this extrusion? The extrusion is modeled like this: that it binds onto to this filament, okay, and each of this end can move along the filament, okay, and hence it can pull out a film. Okay, think about that. I have this thing moving, okay, or binding onto a filament, okay, and I have another string, okay. So maybe I have here, okay. So it is binding here, okay, and so this is my cohesin molecule, and it is binding here in this point, okay, and it is pulling this both sides like that, okay. It is pulling that, moving along that and that, okay. So again, cohesin is so we kind of model it as it's moving along that. There are details of this thing which I will not talk about here, okay, but if somebody is interested, we can talk about the details of the model. For the time being, let's assume that this is moving along the polymer and then pulling a loop. Okay. Okay, and then there are these red spheres here which are marked as CTCF. Okay, and when this loop reaches the CTCF, it stops. Okay, and these cohesin binds and unbinds with the rate R plus and minus. Okay, so there is a rate of binding and unbinding. Okay, so it's not that whenever it binds, it goes to the end. Okay, it can fall off, and there is a rate with which that.
So it always binds to the two nearing bits, or it can bind to also to.
So that is the question, which because see, it is not clear. Okay, but you saw the probability of you saw the P of S, right? Which are very, very small, right? So the probability that it binds to two faraway regions which come very close is very, very small. So we take that as it's always binding close by. Okay.
So the unbind, it can unbind from any.
Yes. Anywhere. Yeah.
Slightly confused. So here you mentioned like how to think about this filament as flexible polymer.
Yes.
Okay. But then once this cohesin is pulling them through,
yeah.
In flexible polymer, due to thermal, the loop will be like very
yeah.
It's a very smooth loop, right?
No, this is a schematic.
Okay.
Yes, this is a schematic. Yeah.
So it is highly fluctuating.
Yeah, it's fluctuating. I'll show you some.
Thanks.
Okay. Okay. So yeah, sorry.
The cohesin is only going to bind in the desired way only if we have a loop, a thermally generated loop.
Small loops, right? Yeah, very small. Yeah.
But the probability of having a loop is smaller than the probability of having, I mean, in a thermally generated thing, is much smaller than the probability of having a loop in chromatin from the experiments. Right?
No. So the chromatin can nearby regions, right?
Yeah, but I'm just saying that the probability of having a loop
yeah.
in this particular case seems to be exactly the same as the probability that a thermally generated polymer will. Because the cohesin is not driving any loop formation.
But here cohesin is driving the loop formation.
Oh, where is it?
Yeah, I'll come to that. Yeah.
No, but it's already, I mean.
No, I'm just showing the schematic. I will show you the thing.
Okay.
Sorry. I'm just showing that it is.
But it's already.
That's later stage.
That's a later stage. This is a later stage. Just showing that it is. Yeah. No, okay. I wanted to show two things here. One is that it can bind, unbind. Okay, that is nearby regions. Okay, that's one. Okay. Second thing is that it can stop at the red bead, which is CTCF, which marks the topological boundary of the topologically associated domains. Okay, that is what the CTCF is. And the third thing is in our model that when two of these things come, okay, and then they touch, okay, they stop. Okay, so now there is some report that they can actually go past each other. Okay, but we have now the model which we have done. This stops the thing. These are the three things I want to show you schematically. Okay. No buckling. This is just a spring connected. Yeah.
Sunil, I have a question. So why each bead you are representing by 2 kV segment, not like some other number? And is it related to persistence length of your chromatin fibers?
No. Chromatin persistence is much smaller.
Mm-hmm.
Much smaller.
Okay.
So this length scale, it is a It is a flexible polymer.
Oh, it's okay.
Okay, right? Why I take 2 kb, I'll tell you in a minute. Because I wanted to model something which is known experimentally,
Okay,
some
fine.
length which is known experimentally, and this is the only way we could model it, not more higher resolution. Yeah,
Quick question. So in your probability map,
yeah.
that would mean that the CTCF contact should be very red.
Very?
Red, because
Yes,
this
yes,
says
it
that.
is. It is.
So you should have like really nice.
Yes,
Okay.
yes, yes. Yeah. Okay, so this is again the model. There's little detail. Okay, so it's a linear spring. Okay, so we showed the beads. The beads are connected by linear springs in this case. Okay, and this is the interaction between the beads. Okay, any two beads. Okay, that's we modeled as a Lennard-Jones interaction between any two beads. Okay, and then you have this CTCF. This cohesin molecule. Okay, and that's like a dimer which is spring connected. Okay, just a spring. It's two dimer. Okay, that's all it is. So other things as much buckling of the thing, etc., is not here. Okay, so now this is what it is. So now it can just say that it can bind, unbind with a rate R plus minus. Okay, and we have a control on how many can bind and how many should be on at any time, or what is the average number of bound cohesin is. Okay, and then once it bound, it kind of extrudes the polymer, the loop with the rate Rs. Okay, so R plus minus is the binding rate, Rs is the extrusion rate. Okay, and so typical time scales of extruding a cohesin is known from experiments is about 1 to 20 minutes. Okay, a typical residence time. Okay, that is this residence time of this cohesin once it's bound. You can vary between 1 to 20 minutes. Okay, and typical extrusion rate is known to be 1 to 5 kb per second. Okay, yeah, 1 second. Okay, and then you should remember that the relaxation time of a 1 MB segment of a chromatin, which is, if you can just think about it as a polymer, okay, and in the nucleoplasm, which is about 100 times the viscosity of water, if you take, okay, it could be of the order of minutes. Okay, so that's what it is. So you can see that if you have 1 MB, if you want to extrude, the time it will take will be similar to the relaxation time of the polymer. Okay, so that is what it is. Yeah.
So why the directionality? Because it could equally well slip off. So there is some directionality, right? That it's
Yes.
the fact that it's extruding and not doing the opposite.
Yes.
Slipping off.
Yes.
So there is something about the linkage or the molecular linkage.
The molecule
there?
itself. The molecule
Okay.
itself.
So
while
that gives it a
yeah,
direction.
so while actually yeah, but this is there are some more details which I didn't talk about here. Okay, it is that the way the cohesin actually binds is there are two binding points. Okay, it can anchor one and move the other one. Okay, and then it can switch. Okay, between these two things. Okay, so it can move both ways, but on the average, it moves in in the extrusion direction. Okay, and it moves one at a time. But here we have more because we are talking about a 10 kb segment, etc. We just take it as that it randomly moves either this side or that side. It's not moving both together.
Switching rate is much slower than the time it takes to.
I'll come to that. I'll come to that. No, it's also thermally fluctuating. It also have other dynamics. Yeah. Okay. So let me move on.
This sort of purely kinetic treatment ignores the fact that there are actually force dipoles associated with.
Yes.
Right.
In
So you
this
don't have any.
What I'm going to
dynamics.
talk about today is the
Yeah,
other one.
yeah,
We
yeah.
can talk about that later. Yeah, this is just kinetics, right? Yeah, there is no. In fact, there is no aerodynamics and there is no other force in this particular model. Okay. So the questions we ask are, how do chromatin interactions and loop extrusion mechanism influence the time taken for reorganization of the 3D proximity of far away segments. Okay, if I start from a random thing, okay, how does it reorganize? Okay, so that is the dynamics of the 3D structure. So that's one question which we are asking. And then how do the relative time scale of this loop extrusion and relaxation effects change your correlation functions? Okay, measurable correlation functions. Okay, and can one rule out this extrusion? Okay, if one sees that static quantities like the one which I showed, the contact map which I showed, okay, is not dependent on cohesin or CTCF. Okay, can we rule out? For example, in the case of Drosophila, a lot of experiments are done, and they look at the contact map and they say that if you just knock out all the CTCF, okay, and you still get the same contact map. Okay, So there's a question that, does that mean there is no extrusion there? Okay. So these are the questions which we are trying to answer. But the dynamics says something different. Yeah.
So this that loop, that dynamics, I can just think of it like a random walk with only on the positive plane, right? It's boundary at zero, right? I guess. I mean, it just goes up and down, but on the average it will go up because I have a...
Correct. Yeah, there is small time, there is fluctuation, but then there is,
it's
like a biased...
0 to infinity domain, if I can think of it.
Yeah, you're saying it's like a biased random walk.
I mean, bias in the sense, it will look like a distinct, mean will be non-zero because I'm disallowing the negative side.
Correct.
And I mean, it will be square root of t, the typical fluctuations.
Yeah, we have not looked at I don't know that detail is known. We know that average it pulls out this rate.
Yeah.
Okay.
Well,
so
it goes both ways, right?
It doesn't go both ways. No.
It doesn't go both ways. But even if you...
Extrusion is one way, right? But a long time.
Even if it goes both ways...
On the average, it's going one way.
But yeah, because no, even if you don't put a bias, if you just take a random walk with a... Like a motor protein. Yeah.
Exactly.
So it's a biased random walk?
In this case, it's a biased random walk.
Biased random walk with a hard wall at the origin, basically, right?
Yeah. So it is moving...
There's a directionality to the...
No, but still also it cannot become less than 0. So there's a hard wall.
Oh, it can't become less than 0. It will fall off. It will fall off. It will fall off because it's binding at nearby region, right? And
no, so the loop is bounded by 0 from below.
Exactly.
Right. So there's a random walk bounded by 0. So it's on the upper half, and okay, there's a bias. But even if you don't put a bias, you'll still have a non-zero mean because you are just it's a random walk on the upper half plane, basically.
Yeah.
Right.
So that was the question I asked, which is, can it equally well...
So, yeah, so on the average, it is moving in one direction. That is enough for now,
Yeah,
and
yeah,
we can discuss the
exactly.
rest of it. Yeah. Okay, so let's move on. Okay, so this is what this is from the simulations. Okay, and what we are showing here. So if I'm just looking at a very weak interaction between the monomers, okay, apart from the self-avoiding thing, okay, it's a very weak interaction between them. And this is my R plus minus rate for binding and unbinding, okay, and this is the extrusion rate, okay. This is kind of some number which is fixed. Okay? And now I'm looking at the contact map as a function of the number of extruders, okay, and this is taken, okay. Now this is a chromatin location, okay. It's given. This is taken from an experiment, okay, on mouse cell, okay. And mouse stem cell thing, okay. So that's what is taken, okay. So that is why, and these CTCF locations are fixed based on those experimental details. And you can see that wherever the CTCF location is there is a hard part. That's what you are asking, right? Okay. Okay. So that is what the CTCF location is, okay. And then you can see that as you increase the number of extruders, you have more contact, which is not surprising, okay. And you can see the configuration also changes. Okay, I'm showing my configuration instead of bead and spring, I'm showing as a tube just for clarity. Okay, and the colors are here represent the different topologically associated domains, the tags. And you can see that as you increase the number of extruders, even though the attractive interaction between them is very weak. Okay, so it is like a good solvent. Okay, but it is still forming a condensed object. Okay? And I can look at that radius of gyration like any polymer guy would look at, have a radius of gyration of this polymer as a function of the number of extruders for different interaction strength. This is purely repulsive, the green one, and then as you increase the interaction, and you can see that if you increase the interaction between them, or increase the strength of the interaction between the new two monomers, and then the effect of the extruder become very weak because it's already condensed. Okay, right? So this is what it is. See. Okay, but.
Loop area distribution?
We not looked at that. No. But there are some experiments which do look at that. Yeah, right. Yeah.
I mean, it seems like the spacing of those red blobs. I forget what you call it.
Sorry.
The spacing of the red blobs.
Yeah.
I mean, because that's the point at which it stops being able to extend
Correct.
any further.
Yes. So it depends.
The distribution of that will tell you what the.
That yeah. Right. So that depends on what part of the chromosome you're looking at, what part of the chromatin you're looking at, okay? And this is taken for a particular case of the mouse stem cell embryo, okay? So sorry, mouse
Sunil,
stem cells.
the epsilon is polymer-polymer interaction.
Yes.
It is not CTCF, cohesin, those.
Yeah.
It's just polymer-polymer, monomer-monomer interaction.
Monomer.
Okay.
Okay, so now I can look at other things. Okay, as I said, this is basically the experiment from which we took taken that. Okay, a specific region of chromosome. Okay, from mouse embryonic stem cell, and that's what is taken here. Okay, and what we're showing that is that the same contact map. If I look at now both CTCF and cohesin are present. Okay, then you get this. Okay, and you can then mark the boundary probability. Okay, there's something which experiments do. Okay, the boundary probability, and you can see the boundary probability here. These are the boundary probability, means the boundary of the topologically associated domains, which you can get from here. Okay, so it's not surprising because there is a CTCF put there, and there is a full stop there. Okay, and now if you don't have a CTCF, if you have only cohesin, you don't see this boundary now. Okay, you see an average contact, because everything is now pulled out and there is an average contact. Okay. And now if you look at CTCF is present but no cohesin present, okay, and then again you don't see any contacts, right? Because now it happens, you know, there is a random walk in the thing. Okay, so you don't see anything. Okay. Now you can compare the 3D distance between any 2 points, because something which experiments measure. Okay. And this is a reference to that, okay, where they actually measure this thing, 3D distance, okay, and it's a hard experiments to do to measure 3D distance between 2 points in the chromatin. So there are not many points available. So these are the points available from the experiments, okay, and then you can kind of tune your parameter, okay, and show that, you know, if you have both cohesin plus and CTCF plus, you can get, you know, within a TAD contact to be higher than between TADs, and they're close by, but if you knock out one of them, okay, you lose that resolution. You see that within TAD contact and between TAD contact is the same. Okay, and so if you want have a hierarchical contact thing, then you would need, you know, these domains. That's what this experiments are telling us, and that's what the model says, because model we have put in that. Okay, but this is what the experiments are saying. Okay, so that is this. Trying to basically look at, that's the reason why we took this particular region, because experiments are available. Okay, and our coarse graining, even though our coarse graining is not specific to mouse embryonic stem cell, okay, but we took the 10 kb region, 2 kb region as one bead, because we wanted to be able to simulate this particular region. Okay. Okay, so you can plot the 3D distance, and this is again just summarizing what I just now said. Okay, and you can show that for these parameters, okay, this is a residence time about 200 seconds, okay, extrusion of 1000 base pairs per second, okay, and we have 5 extruders, okay, and you have an epsilon of 0.3, and you can show that between the TADs, this is the experiment and that is the simulation, okay, and this is the experiment and that's the simulation, okay, the 3D distance distribution, okay, and you can see that you can kind of match it, okay, if you have both cohesin plus and CTCF. Okay. Otherwise you can't. Okay, so let's go. Okay, so it is known that, as I just now mentioned, that Drosophila, that CTCF loss has very limited genome limited effect on the genome architecture, which means on the contact map. Okay, very limited effect. It is known for the Drosophila. Okay, so we also say that if you have higher interaction between particles, okay, epsilon equal to 0.4, okay, and then if you have cohesin minus or cohesin plus, okay, you are not finding much thing. Okay? Higher interaction between 2 points within a TAD. Okay, it's like, you know, your TAD is now defined by some other chemical modification. Okay, and then you have very high interaction within a TAD. Okay, and then you will not have any effect of cohesin extrusion on that, because what is dominated is a direct interaction between the thing. So this where the condensate forms, that kind of stuff, okay, where you have, if you have that kind of regions, and then you will not have effect of extrusion visible in this structure.
Without the CTCF, what sets the domain size?
The interaction. So I showed you earlier, for example, some condensate can form, right? That kind of modification of a. So now the TAD is not defined by the CTCF, it's defined by the modification of the chemical.
Like these boundaries are not fixed. These are like some emergent.
No, it is fixed by chemical modification of the of the filament itself. Okay. So that's what it would see. Okay. So that could be one possible reason. It's not clear. It's a reason, but I'm saying that if you have something like that, okay, and then an extrusion will not be visible in the static structure. Okay. Okay, we'll come back to this thing again. Yeah.
Sorry, so is it okay to think of the cohesin CTCF combo as staples on your polymer, right? Why do the distance between the staples also decrease when you are not changing epsilon? So when the epsilon was fixed,
because you pull the loop, no?
No, but once the loops are pulled, so now you should have a static picture of all the loops.
But the loop
being
is not pulled permanently. The loop is continuously pulled and.
Oh, so that dynamics goes into this, is it?
Yeah,
okay.
Yeah, I'll come to that. I hope I get time here. Okay, so let's go on this. Okay, so this is saying, okay, now I put an epsilon into a 0.4. Okay, rest of the things are same, but I put 0.4 here, and already we don't see any more effect of the CTCF here. Okay, so that's about the static structure we just showed. Okay, now what about dynamics? Okay, so I wanted to spend a few minutes on the dynamics. Okay, and so this is what you're looking at. Okay, so again, just to remind you, okay, you have a DNA. Okay, you have the CTCF sitting at the two ends. Okay, and you have chromatin, so this loop extruder binding onto it. Okay, cohesin. Okay, and then it moves by pulling the loop. Right, there's a one-dimensional representation of that. It is pulling the loop. Okay, and then you can have, depending upon what is the rate at which it is moving, the RS value, where the loop at which the rate at which it is pulling the filament, okay, and the amount of time it stays, okay, you have what's called the processivity, right? That is RS by R plus minus, okay, that defines the processivity. Okay. So low processivity means by the time it moves a little bit, okay, it can fall off. Okay, high processivity means it can move much more. We can pull a bigger loop. Okay, so high processivity is bigger loop pulling, and low processivity is smaller loop. And you can control that by either changing the residence time, okay, the loop extrusion speed, okay, or by the rate at which it binds and bind. Okay, both are controllable. Okay, now there have been some saying that processivity only matters, only the ratio matters, it doesn't matter what is the residence time is. Okay, what matters is only the speed at which, the processivity only, the ratio only matters. Okay, so that's something which we wanted to see in the simulations. Okay, so again, just to remind you that speed of extrusion is from 1 kb to 5 kb. Okay, and per second, residence time can vary from 5 to 20 minutes. Okay, and relaxation time segments can vary from 3 to 15 minutes depending upon the size. Okay, 100 kb of mega base pairs. Okay, so let's look at this extrusion rate effect. Okay, so here I'm saying something. All parameters, the processivity is fixed. Okay, so I fix the RS by R plus minus ratio. Okay, and I look at low residence time versus high residence time. Okay, same polymer, and I'm just showing you the thing. Okay, what happens? Okay, and this is the kind of steady state the thing. Things are binding, unbinding, extruding. Okay, and at the same time, there is also thermal relaxation of the polymer. Okay, and you can see that we have low residence time. There is more binding, unbinding happening. Okay, the polymer is more condensed. Okay, and if you have a high residence time at the same extrusion rate, okay, so the processivity is the same for both of them. Okay, and you can see that the polymer is more relaxed. Okay, and then it's much more bigger. Okay, so that is what you see. Okay. So there is an effect of this time. It's not just the ratio which matters. What matters is also residence time, okay, because there is a fixed relaxation time for the polymer. Okay, so now I can look at that, the radius of gyration as a function of processivity for different values of residence time, and you can see that as you decrease the residence time, the polymer becomes more compact. As a function of processivity, of course, everything comes down. Okay, but as you decrease the residence time, the polymer becomes more compact. Okay. So this effect would be seen in experiments if you look at it. Okay, that's what one of the suggestion is. Okay, so one of the things experiments which people do is to look at, as I said, they measure 3D distance. Okay, they can also measure 2-point correlation functions. Okay, so they can look at this vector, okay, and look at its correlation function, okay, as a function of time. Okay, and that is so then we would expect a 2-point correlator in the case of to show a power law exponent as a function of the distance, the arc length, right? Okay, that's the power of gamma, and Rouse predicts 2.2 for self-avoiding random walk, okay, and 5/3 for a fractal globule. Okay, these are known numbers from polymer physics. Okay, now this is what the experiments show. Okay, this is Buckner et al. Okay, this is in 2023. This is the experiments show. So they show the relaxation time of 2-point correlator as a function of the distance arc length between them, or the genomic distance between them. And this is what they see in the experiments. Yeah, and that's what the ideal would predict. Actually, is about to be about 2.2. Okay, and because it's a self-avoiding random walk. Okay, and the crumple, this fractal globule should go 5 by 3. Okay? But this shows much smaller. That's what the experiments show. Okay, so the main message of this paper was that none of the polymer models are predicting this correctly. Okay, so and what we show that without extrusion you would get that. Okay, as you increase the interaction between the monomers, you can get different exponents. Okay, of course, because you will go into from a self-avoiding random walk in a good solvent to a bad solvent behavior as we increase epsilon. Okay, so that would change. Okay. But with extrusion, okay, if you have same a number of extruded as 5, you can see that for different things the exponents are very different with extrusion. Okay, and you can actually get even 0.7, and that's what this exponent actually is. Okay, and then and you can, for a fixed value of epsilon, again changing the number of extruders also changes the exponent, not very much, but it changes a bit. Okay, but 2.2, 2 for Gaussian polymer. Okay, and if you self-avoid, yeah. Okay, so this is what our dynamics would show. Okay, so that with extrusion you can get very different exponents. Okay, and basically it is not universal. Okay, so depending upon what extrusion rate is and what is the number of extruder is, okay, and what is the interaction between polymer is, everything is, you can get different exponents. Okay, so this is. Huh?
I mean, it's only over 1 decade, so when shouldn't you talk about experiments?
Yeah, we don't know, but that's for the only experiments we have, right? Yeah, so maybe people will measure it over over time because it's rather recent. Yeah.
It's quite well known that it should
Yeah.
not.
What is no?
I mean, there's no scaling behavior.
No, there's no scaling behavior here. I'm just saying, yeah, so that is true. Yeah, there is no, yeah. We are looking at the same size as here. Okay, we are actually looking at the same size here. That's all we are looking at. But that's all the experiments we have now. Probably we'll have much better thing here. But what we're going to show is that say that because of this extrusion, you don't really expect to be having a universal scaling anywhere in this okay, because it could vary a lot, right? Yeah. Okay, and so again, so we say the low residence time. Okay, low residence time, different extrusion rate, you can get different exponents. Okay, if you call them exponents. But the most interesting thing here I want to show is that not really what are the scales is that if you have low residence time, you can get different values for different extrusion rate. But if you have high residence time, then they all go the same way. Okay, they just collapse onto one another. So they have hardly any effect. Okay. So again, want to show that it's not the processivity which matters; it's also the residence time, okay, of what matters because of this competition between these two time scales. Okay, that's what basically we were trying to show in this paper. Okay, so the question we can ask is extrusion present in Drosophila, because we saw that the contact maps doesn't show any effect of extrusion. Is there anything which is which the dynamics can show? So we are just saying that if you look at simulations, okay, and take the same interaction which we took, for which we got the Drosophila kind of dependence for epsilon equal to 0.4, okay, plus my cohesin minus cohesin, okay, the relaxation time could be very different, okay, and they will show very different scaling, okay, very different behavior, okay. So yeah, so you could ask the question that whether there is actually a linear regime here or not, but that is we don't know, but we can see that there are very difference in the behavior as you plus cohesin and minus cohesin, okay, and that should be clear from the experiments, okay. But people have not really looked at this in the experiments yet, except that one paper which we have, okay. Okay, so that is what, okay, now I can also look at single point relaxation function, okay, and again experiments show that single point relaxation function is independent of everything, okay, and that's what we see here also in the simulations, okay, or the extrusion, without extrusion, okay, residence time, etc. Single point relaxation function is always showing you about 0.58, okay, so again just showing you those dynamics. Okay, so that is what I want to do. So I don't know whether I have time. Should I stop now, or I can take 5 minutes more. I might need another 5 minutes just to show something. Yeah. Okay. So I wanted to show you. So that is about the loop extrusion. Okay. Now that is at a larger length scale we are talking about. As we talked about, minimum our resolution was 2 MB. Sorry, 2 kilobase pairs. And now we talk about nucleosome resolution. Okay, there is also other things happening in nucleosome thing, right? So you can, as I said, these are octamers on which it's bound. Okay, and you have this nucleosome tails, or the tails which are projecting out. Okay, and you can change the chemical state of this tail, and then alter the way it is bound. Okay, and it is known that acetylation would actually loosen this thing. Okay, loosen this binding. Okay, acetylation of this tail. Okay, and deacetylation and methylation would actually increase the interaction between these things. Okay? So when you acetylate and in the presence of other remodelers, okay, it can actually fall off. Okay, there are other remodelers, okay, like RSC, etc. So which are again ATPases, okay, which can actually act on this when it is acetylated, and it can actually fall off. Okay, and you can change the distance between the nucleosomes, and you can make it dynamic, etc. Okay. So we wanted to model this. Okay, we had done this earlier. So we wanted to model such behavior. Okay, and we okay, before that I'll say some really recent experiments. Okay, which has just come out this month. Okay, in which they look at exactly this. Okay, you have the nucleosomes. You have this condensates. Okay, the chromosome condensates. So these are in vitro experiments in which people actually look at a finite string of nucleosomes. Okay, they can form that nucleosomes. Okay, and then they form a condensate. Okay, and then they add ATP, and then they add RSC or ACF. So ACF is a slider. Okay, it actually slides the nucleosome. Okay, along the polymer. It doesn't unbind. It slides. While RSC would kind of unbind it. Okay, it can evict the nucleosomes. Okay, and also it can just big molecule. It can actually bind onto the nucleosome and shield the interactions too. Okay, so there are two effects in this. So these kind of experiments are there. Okay, so it's coming up now. Okay, in vitro. Okay, so I'm hoping that it will be quantifiable. And there is some movie which is interesting here. Okay, even though I don't have an answer to it, but I thought it was an interesting movie. So if you have RSC added to the condensate, okay, and before adding RSC, okay, this is ATP plus the nucleosomes, okay, and you can see this nucleosome condensates here, okay. These are this chromatin condensate, okay, and the chromatin string condensate, I should say, okay, and that's what is being shown here, okay, and I believe that what is fluorescing is the nucleosome, okay. So you can see that. Okay, and then if you add now RSC to it, and then they move around quite a bit, and they lose their spherical shape. They actually even become bigger. Okay, their data shows that they become a little swollen. Okay, and they lose their shape, and then they move around. Okay. It's not clear why it's happening, but they do move around. Okay, and so what is happening here is binding, unbinding of this RSC, and eviction, and also reforming of the nucleosomes, and that seems to lead to this kind of motion. Okay, so we have a earlier model, and we're now trying to use the model to kind of model these condensates. Okay, but I will not go into that. I will just show you that model. So the model is that you have nucleosomes and DNA wound around it. Okay, and you can have these nucleosomes for them falling off. Okay, and then hence there is distance between them changes. Okay, and they can again bind, and then the distance between them again come together, and this can also, and there is an interaction between nucleosome and nucleosome. Okay, and this can be modeled as just having a segment which is think about this polymer as a block polymer. Okay, a block copolymer. Okay, where you have regions which are in good solvent and versus bad solvent. Okay, so these condensed regions we call as in bad solvent, and extended regions as in good solvent. Okay, and you can have this block copolymer, and you can switch them between these regions. You can switch between the blue and the red. Okay, and then you can get them to deform in the thing, and then you can just this is a model. Okay, you have, red beads and blue beads, which are solvophobic and solvophilic, and you can switch between these two regions. Okay, there's some time scale. Okay, and then you can look at the dynamics of this. Okay, so I did not go into the details of that. I just wanted to show you what happens if you do that. And if you look at the radius of gyration as a function of the time scale it which takes for it to switch between these two states. And so you're switching between a condensed region and expanded region of a polymer. Okay, and then you can see that if the switching time scale is very fast, you can actually get the polymer to swell. Okay? And the switching time is very small, then it actually condenses back into it. Okay. So the polymer size can actually vary as you change the switching time. So we believe that there is some relevance to what these experiments show. Okay, and we can show that this switching time is, if you match the switching time to segment relaxation time, then you get a maximum swollen state. Okay, and if your switching time is same as the polymer relaxation time, you get the most condensed state. Okay, and which is similar to what the extruder also showed us. Okay, and this is just a movie of that. Okay, where you see that this is a switching time. Okay, this is a very fast switching time. The polymer looks condensed. Okay, and as you increase the switching time, that means it becomes slower. Okay, and the polymer would kind of swell. Okay, and then when you go into higher and higher switching time, they condense back again. Okay. So yeah, so then now it will condense back again, and then it will finally stay as one globule. Okay, so we were trying to use this model now to also look at condensate of many such polymers. Okay, and then look at what is the dynamics of that. This work is done with Monte Carlo, but now we are doing is molecular dynamics, including hydrodynamic interactions to see what is the condensate behavior of such polymers would be. I think that's where I wanted to stop, and yeah, I think that's what it is. You can take any questions. Right. Thank you. Yeah.
Length of the chromatin
So that can vary. But chromosome lengths can vary from 50 MB to 250 MB in Indian for a human chromosome. I think 23 or something is the maximum. I think 250 MB megabase pairs, and the smallest, smallest is about 50, 60.
No, what I am not understanding is the probability of contact is very low. But if these are packed very tightly, why it is so low?
Because it's also dynamic. Yeah. There also, it's a very crowded environment. You should also think about this. It's also a very crowded environment. Okay, there are many things happening. Okay, and so yeah, so it's also very dynamic. Okay. So it doesn't stay connected all the time.
Oh, okay. So you will say contact only when they are staying.
Right. Correct. So the way the experiments they do is to add some linker, right? Okay. So whatever is in contact will get linked. Okay, and then they slice off those linked regions, take it out, and then they delink it, and then they sequence the thing, etc. Yeah, that's how they do it. Yeah. Right.
In the chromatin folding within the cell, what is the role of ribosome? Does it play?
No, ribosomes are only to read the. Yeah. So ribosomes are later. Okay, ribosomes come later. Okay,
Yeah.
here.
It has no role in the.
No, not inside the nucleus. No, ribosomes are outside. Yeah, yeah, outside the nucleus. Yeah, yeah. Okay.
Contact is low in 3 dimensions. Contact is low in 3 dimensions.
Yeah.
2D
No, it's 3 dimensions. Yeah, yeah.
I just want just comment.
Yeah.
The contacts are actually very high because you have to compare it to something is much higher than the random.
Higher than the random. Okay.
No. This goes so the random goes as 1 by s to the power 3 by 2.
I know.
This one goes as 1 by s.
But it is 3D. But it is 3D.
Yeah.
Random or then it's transient, like it doesn't come there. If it is 2D,
No, 3D is. Yeah.
3D goes as 1 over s to the power 3 by 2, right? So, no, 3 by 2.
Yeah, exactly. That's the point, that there are actually active processes.
It's in a potential.
Yeah.
Then it's exponentially likely to.
So it's a question, when I say low, it's question is compared to what is the question, right? Yeah, okay. Yeah.
So, Sunil, I was just thinking that the first model that you have, I feel that there's a way to map it onto, Mustancer's model on passive sliders. But if you do that with the French randomness, I think we have done certain level, we have done. The model that you have gets mapped
Yeah.
onto that, and that predicts a non-equivalent phase transition. That might be interesting too.
Yeah, we did look at it once. I did look at it once, but there was something I don't remember exactly now what it is.
So you should look at the passive
Yeah.
slider model in French randomness.
Right.
That corresponds to the CT slides.
Yeah. Yeah, I did look at this. I'm aware of this work. Yeah.
Sunil, for a different ambient temperature, the T is smaller, then would the contact property go up?
I don't know that people have done it at different temperatures. I have no idea. Yeah, I'm sure there should be some effect of temperature, but I don't know.
So can you, in the process of DNA replication, whether you want mitosis or meiosis,
Yeah.
whenever there's replication, you need to unwind everything,
Yes.
right? So in that process, what role do these those
Nucleosomes.
sticky molecules, those little
Nucleosomes, right? It's an
those
interesting
little
question.
two little cohesins
No, cohesins and all fall off at that point. of
fall
time.
off, and
Yeah.
then the whole
Yeah,
chemical
right.
process.
Correct. Cohesin has some role when after chromosomes condense and binding them, etc. But that as a binder, not as an extruder. Yeah, but
So all of these guys become not important chemically, not
Yes.
important, and
but
some
when the
other
replication happens, the polymerase reads it off, right? And it reads it off, and then it produces copies. Okay, the DNA replication happens, but then the set of nucleosomes, right? So that can go to only one of them, right? But there are nucleosomes, there's histones available in the complex, because histones are also doubled at that time during the S phase. Okay, but then it cannot go into both. It can go to only one of them. So it's not very clear how that happens. Means how does it retain the same epigenetic thing? It is not.
Stones also, these things that they're wrapping around,
Yeah.
they also are there in this process, and they also get shared.
Yeah, that's what I'm saying.
That's the
So
interesting.
they go
okay, I
to
see,
one
okay.
of them, right? One histone complex will either go to one copy or the other copy.
So you're saying that they will replace.
But then there is some way they build up that thing, same thing. So it's not clear. Yeah. How it happens, it's not very clear. There are some models for it, saying that if you copy only every other one goes to other one, that's enough to keep the information, etc. Yeah.
Yeah. So again, it's a matter of detail. So that CTCF bright dot that you see in your simulation, correct? But you don't see that in the experimental data, because we would know where the CTCFs are.
Yeah, you do.
So in that experimental data that you showed,
Yes.
there are CTCF bright dots.
Yes. Yeah. You see that the boundary probability which I showed.
Yeah.
Okay. So actually the same thing which experiments shows. So that comes from the CTCF bright spots. So that's not. Okay, it's not. See, in our case, the CTCF is a hard stop.
Yes.
Okay. We can modify that a bit, and we can say that the CTCF is slightly spread out. It's not one spot. Okay. One spot means we are talking about here 2 kb resolution. Okay, we could say that it's a little spread out. Okay, but then that bright spot will also get a little diffuse. That's all. CTCF, yeah, it can bind and unbind, but there's not one CTCF complex. There is a series of complexes. So overall, there is a stop.
But
that
CTCF
stop
is
is not at one point. It is spread out.
But CTCF is a very small region, like 2 kb is much larger. So it is well within that 2 kb, one spot.
No, I'm saying that it will not be. Yeah, it's exactly. It's well within that.
So it will be like 1 pixel that should.
Yeah.
Right.
If it is, it depends on your resolution, of course. Yeah, right. Yeah. If you're taking as I showed that thing about 30 kb resolution, then it will be just a, pixel matter thing. Yeah.
Yeah. So one point of time you mentioned about filament severing. So do you consider that in your dynamics? The filament can cut because why knots are
Yeah, but we are simulating much smaller segments in these things.
Okay.
We have much larger segments, then you could have entanglement. Okay. And we are saying that entanglement is not relevant for
so
knots
there
are,
can be abrupt
knots are not
relaxation
relevant
due
for this
to-
because you have your dynamics is not limited by knots or subforming, etc. Because they can cut and rejoin.
But can it cut even smaller segment, or that is not
It
relevant?
can, but I'm saying that kind of constraint doesn't appear in these simulations.
Okay.
Because these are smaller.
Because that can affect the relaxation and all, right? Like
Yes.
in presence
Yes,
of.
if it's longer one, yes. Yeah.
Oh, right.
Yeah, because even at this scale, you had things being pulled up for your this extrusion, but you had a knotting from another part of the. Even at this scale, if you have this extrusion happening, and you have knotting, it's pulling it in the other direction, right?
Right,
That's
right.
kind of a dynamical knotted system, which is topologically non-trivial. You can't, and it would change the dynamics of the extrusion.
That's
So,
right.
but it depends very much on.
Yeah. Yeah. So basically, we're saying that if you are really looking at, if you're saying whether self-avoidance is important or not, because right, if you're saying they can cut and rejoin all the time, okay, then self-avoidance is not important. They can go through
Even
one.
at this scale, they can do that.
They can do that, right? Yeah. They can do that. Yes. Yeah.
Sunil, what was your answer to the question of bias for this cohesin? Why does it prefer one direction to the other?
But that's how the motor is. It moves.
I
in
know,
one
but
direction.
what is it? Is a polymer, the polymer
No,
doesn't
no,
have
polymer
any
has
direction.
no, it's a motor.
Why? Because it's a sequence of DNA, I mean, so nucleotides.
The DNA has a direction.
That is true at the ends, yeah, okay. That is what is giving it.
It's walking along in one direction.
Yeah.
Like condensing,
Right.
on the molecular motors.
So one head of cohesin prefers one direction and the other head prefers the other direction?
Both. Same
Sorry?
direction.
It's not.
That's So with the shorter time, apparently it moves one of them at one time, and it can move both ways. But on the average, it moves in one direction.
How does it move? I mean, locally, the direction that
Well, there's a sequence, but I don't know if that sequence matters or not. I'm not sure. My understanding was that the motor itself is designed to move in one direction.
The track on which is moving is polar, just like there's
Yeah,
a
but
plus
that's
and just like actin or microtubules.
Right, yeah.
Yeah, but it's the
And it's that provides.
phase, no, now.
Could be, yeah.
Moving this, the other one will go slightly.
Yeah.
So that will not make a loop, right?
That's also true.
No, you're saying it will move the other way.
Yeah, because if this guy moves this, the other guy also moves.
But how do you know the other guy is identical?
I don't know,
They're all
but
different pieces. So obviously it won't do it for symmetrical objects.
so like
Right.
in your, so basically this control length of the this thing that increases linearly with time or with the square root of the,
I don't know that people know it.
that's difficult, is it, experimentally?
I don't know. I don't know that people know you're saying the loop length.
Loop length, if it is linear,
Average-
that means there's a bias.
I
or
know
if
the
it is-
average
Huh?
is average speed people know. Okay, average speed people know. But now whether people have really looked at what is the rate at which it is going, what is the scaling of the specific time, I don't know. I've not seen the data. I don't know the data.
Those speeds are all identical.
Is it no? So what is the data? I don't know. Huh? In vitro only, yeah. It goes linearly. Yeah, it
It's
goes-
linear, is it?
yeah. I've not seen it.
I see.
Oh, Mahipal's work, right? Yeah. Okay.
So then I am asking also, may not be relevant. So this is packaging. Now, if somebody, when it has to read the DNA again, you have to unpackage it.
Yes, it loosens. That's what I'm talking about, the acetylation and deacetylation. Okay, so if you acetylate the histone tails, then this packaging becomes loose.
Okay.
Okay, and then polymerizers can actually read that.
And so in that loop extrusion will opposite direction it will work or no? No, that
Loop
time-
extruder is acting at a much bigger scale,
Okay.
bigger length scale. Okay, you're talking about at the nucleosome level.
Okay.
This is what happens. Is the acetylation loosens the binding, okay, and hence polymerase can move on it and actually read off the thing. So that's one of the things, with the regions where the cell doesn't want the DNA to be read, they are methylated, okay, and then they are condensed, okay, and then the polymerases don't read.
Okay, then if there are no more questions, then let's thank Sunil once again.
Thank you.
And it's a token of appreciation from RRI.
Okay, thank you. So somebody has to take a picture, okay? Okay, no. So Pramod, I want to take a picture with you on this thing. So let's take it. So what is this stuff? Okay, it's a book. Yeah, please come. He will take it. He'll take the picture.
Maybe you
Okay.
come this side.
Okay. Thank you.
Found these things. I was not sure. You send it to my